quickly, not recording, now we're recording, now our class is underway. All right, and so I actually just got the link set out this morning for the Tuesday lecture that the projector was messed up. And it's working now. I got over here early, and here we go. So, because we have a test in a week. And so we're going to finish talking about chapter 19 today, and then we're also going to have chapter 20, because our exam is going to cover chapter 18, 19, and 20. And it'll be the same style format as the first exam. I'll try to write it in a way that you'll be able to get done in time, but you'll have to be busy, and the questions will be hard, like the first time. But I'll try to do that. But you also will need to come, with just the right amount of caffeine in your system to be able to work, just work and get your answers down. And remember that sometimes the answers are pretty direct. And if you know the direct answer and you're confident, it could just be like a list of just 3 words on some of them. And I intentionally do that, where many of them are really short answers. And then there's some where I ask you to do more, a little drawing like that, stuff like that, okay? And understand that sometimes your answers are going to be long, and that's absolutely fine. You just be mindful of that and how you work and get through the exam. So chapter 20 is a really satisfying chapter for us. There's new information, but it's really using the principles that we've been talking about since day one. Then I'll say something about the term paper on the next thing, and then we have our exam, okay? So the next thing is, I apologize, I was confused about the due date on the syllabus. Here I say, for the term paper, it's fine to me if you get it to me by Monday the 24th. And when you're done with it, you can email it to me, and you can keep asking questions about it and stuff like that. That will allow you to work on it the weekend after our exam and finish it up. Because everybody wants to focus on the exam right now, like that. I guess when I was making up the syllabus, I was thinking, let's get that done before the exam. And then I was thinking, we should finish it up after the exam. And so let's finish it up after the exam. You already handed it in, 2 students did. You could send it again if you want to work on it more. If you just didn't get as much done on it as you wanted, you can work on it more. Just let me know. And so again, I apologize about that. All right, so here we go. Do people have questions about anything they want to ask about?
Yeah, but you need to figure it out.
Okay.
Just to figure out, I know I had a question about something, but
okay.
I can't remember what it was.
So I just want to show a couple of slides that I would have shown on Tuesday. This is one we're familiar with. Here's a cell that is going through apoptosis, and so it has had the inhibition of the caspases released. So the caspases are always in our cells, and they're always inhibited, and apoptosis involves the release from inhibition. BCL2 is the most famous inhibitor, and it's in our cells inhibiting caspases. So we have to release the inhibition. That can happen either internally. The cell can say, I've got too much damage, I'm not repairing it, and so P53 is encouraging me to activate the apoptotic pathway. Release the inhibition, activate the caspases, and have the cell die. Or it can happen externally from an external factor. That's what happens to those cancer cells that Riley's been teaching us about, is that some external factors are saying, you're going to have to die, and we're going to eat you, like that. Yep. And so what happens is the caspases get activated by a regulatory pathway. Now they're active, and they are proteases, and there are several of them that are numbered. We don't need to detail the numbers, and they do their job. And their job is to specifically clip parts of target proteins, and one of them is scramblase. And scramblase is present in our cells right now as an inactive pre-protein, a protein that's made and is just a little bit longer than it should be, and as a result, it's inactive. And caspases just clip it, now it's active, and scramblase's activity is to flip phosphatidylserine from the inner leaflet to the outer leaflet of the phospholipid bilayer, and receptors on neighboring cells, especially immune system cells, get activated, and they phagocytize. They ingest the dying cell and recycle its parts. So that's one of these, and what we talked about is it'd be great for us to know some about that one. Then we talked about some about this one. The caspases also clip a part of the protein for a deoxyribonuclease off. The deoxyribonuclease or DNase is a preprotein, inactive preprotein. It's clipped, it's activated, and it just starts cutting the DNA out. And that's a good one to know just because you're saying, well, a cell can't be alive if it doesn't have its DNA. And the answer is yep. And then the third one is there's some mitochondrial involvement. The mitochondria are going to become defective, the cell can't live, but they're also going to release some factors. And here's the visual from the book about the DNA getting broken down. And here's a visual from the book about the mitochondria being activated. And the mitochondria gets activated and it releases factors that can help overcome the inactivation of the caspase and lead to apoptosis. So there are really two roles the mitochondria is playing. One is for it to become inactivated, but the other is to release specific factors. You can think of the mitochondria as a bag holding these factors safely away, always ready to release them. And it says, like, if my activity is going to be disrupted, I'm going to release these things because we shouldn't continue. Yep. Yes.
I just had one question about that, because mitochondria were uptaken by, like, they weren't part of the original cell.
Yep.
And they were uptaken by the purple non-sulfur cells.
Yep.
So, like, was this pathway present, like, within the mitochondria that it would do this prior to the uptake? Or, like, did this develop as kind of like a or evolve as like a kind of stimulatory thing?
Yeah, that's a good question. Here's what I don't know very much about apoptosis in single-celled, modern day single-celled eukaryotes. It's very important in plants and animals, like when a plant dries out in the fall, like that. That's a lot of programmed cell death going on there, and then lots of programmed cell death in animals. I just don't know. So that would be part of the answer as well. Do single-celled eukaryotes feature this? Is it really an integral part of the, that pathway? So I don't know. Just want to point this out that in this pathway, some of the factors are pro-apoptotic BCL2 family members, and they're binding to the mitochondria. So that's a role for pro-apoptotic factors. That family is complicated because, just show you this, here's the easy thing to remember about that family. Here is an anti-apoptotic factor, so it's like BCL2. It's inhibiting programmed cell death. So that could be in BCL2. And some of the pro-apoptotic factors just bind to it directly. So that's the easiest one to remember, is here's BCL2 and it's inhibiting the caspases, and another one just binds to it and blocks it. And last time there were questions at the end, and I say, we're going to inhibit the inhibitor, which is confusing until you see a visual like this, and you're like, okay, so there's the inhibitor, that's always present, and we're going to have some guys that are pro-apoptotic that are just going to work by inhibiting the inhibitor.
So inhibiting the inhibitor leads to more growth?
No, death.
Death. More death.
Yep.
Okay.
Yes, BCL2 is an inhibitor of apoptosis. It's always in there. And that's what we should think of. When we think of apoptosis, we should think of, it's remarkable that all of our cells have caspases in them right now. And they're the effector that can kill the cell. The caspases are inhibited, most famously by BCL2. Not always.
Okay.
Not in every cell, because it gets complicated. But for us, to introduce this topic, think of BCL2 as inhibiting the caspases, and the most common way for us to cause programmed cell death would be to overcome the inhibition, like that. And the pro-apoptotic factors, like the ones we didn't learn the names of, but other family members, do that. This just gives you a little hint, a little show that This story's more complicated. There's a lot of stuff going on with the BCL2 gene family members that allow them to be inhibiting programmed cell death or activating it. Anti-apoptotic or pro-apoptotic. And that's a detail, but I just want to show you that here. That's one of the take-homes from this. And the other take-home from this is that there are actually things in the mitochondria right now that can be released to help activate the caspases.
So backs and back, they bind to the mitochondria and then help break it down?
Yes, and I'm trying not to even say their names because we only one name we need to learn is BCL2. And then these others are BCL2 family members. They're paralogs of BCL2. Some of them are anti-apoptotic, some are pro-apoptotic, and then you just get down into the weeds really fast and it just becomes really confusing. So I just want to show you that just to show you a little bit, but it's the detail. We have much more important things we want to get out of this stories. Yep.
So BCL2 gene family is usually like inhibiting apoptosis, or not always if they're
So it just,
pro-apoptotic.
yeah, let me just show you this visual because we couldn't see it last time.
I guess for like the
Yep.
definition, I can just look this up.
Yep.
real quick, but-
So it's really easy, right? Like this, where we say, Dave has a brother and sister, Craig and Bart. So Dave is in a family. And we can talk about Dave, BCL2. We'd say BCL2 or Dave. Or we could talk about Dave's family, right? And those are two different things, and it's based on the word choice we use. We either say Dave in our answers on the exam, we say directly Dave, BCL2, or we say Dave's family, the BCL2 family. So that's the distinction, is you have to use the two word choices, BCL2, BCL2 gene family, okay? BCL2 is always an inhibitor of apoptosis, and it's one of the most famous proteins in our body. It's super famous for inhibiting apoptosis because it can get mutated in cancer and fail, or no, and keep inhibiting apoptosis when it shouldn't. Okay. Then we have the BCL2 family, and that's where we just gotta breeze through and say, well, some of them are pro-apoptotic, some are anti-apoptotic, and it gets really complicated, right? Like that, right? Like Dave's family. Craig is just a goofball, so that gets complicated because I'm not a goofball, right? Or Barb is really serious, right? You're like, okay, all right, you know, like that, right? So BCL2, anti-apoptotic, BCL2 gene family, both stimulating and inhibiting apoptosis.
What would you say, like the functions of pro-apoptotic things do?
Yep. So there are two ones that we can see here, and they're nice to see. One is here's the BCL2, and here's one like BAD or BIM or BAX or PUMA, I don't know. And it's just pro-apoptotic because it is blocking the anti-apoptotic.
That's like,
That's one.
okay.
And that's great. That's the inhibitor of the inhibitor, the double negative, right? And then here's the other one I just showed you, just to get you that visual. Another pro-apoptotic pathway is just to start binding to the mitochondria and disrupting mitochondrial function and causing the mitochondria to release factors that help activate caspase. There's a lot more, and again, we don't need to dwell on it. Okay? What we want to dwell on is the BCL2 sum, because we want to talk about that. Let me just show these slides because I want to show them last time. But we're talking about paralogs right now, a gene family. And paralogs are two genes in the same species that share a common ancestral gene. So we have the BCL2 gene family, and they arose by gene duplication of an ancestral family member, like that. Here's a nice visual of that. That's what a gene family is like. It's like a set of screwdrivers. There was one ancestral screwdriver in a simple organism that could get everything done for that organism. Then as organisms evolved complexity, they got some gene duplication, so they have more sophisticated toolkit. Right? One of these is BCL2, like that. Right? You could think of it as ancestrally, maybe you had a screwdriver and it could only turn the screw in. And in a more complicated setting, you'd be like, sometimes we want to turn the screw out. So we got some screwdrivers that turn it in, and we have completely different ones that turn it out. That's what the gene family is like for BCL2. Okay? So then, we also want to talk about orthologs, because here it shows fruit flies and mammals like us, and then a model organism where these were first discovered, a little worm called nematodes. And they were discovered down at MIT in one lab, and they won a Nobel Prize. And they had some funny names over here, because they were discovering them, and we're learning this name, BCL2 caspase. Don't worry about it, but look at all these orthologs in here. It's just a really complicated regulatory story. It's complicated like the regulation of START was in chapter 18. And we dove down into that because we really knew a lot about it already. We're just not going to go there here because it's not that worthwhile with that, okay?
Do you want us to learn those names?
No, just two, BCL2 and caspase. Those would be great names for us to know. You can see here that, we got the three talked about before. Here's an activator, activator, and it's just complicated. BCL2 actually inhibits, not directly the caspases, but it inhibits an upstream activator of the caspases, right? And we're already sufficiently confused to not worry about the details. Because look at all these arrows and all these blocks, because those are inhibitors and those are stimulators. And it's just a bunch. Look how much more complicated it is in a big, complicated organism than it was in this little simple worm. Right? Just clink, clink, clink. That's the whole story in that worm. It was very easy to discover. That's a really important principle in molecular biology is go to the simple worm and figure out what's going on because we're closely related to it. And we'll find these orthologs that are remarkably closely related to it. And learn a lot about our health by studying these simple worms. So that's a really important visual. The other is just to notice that fruit fly people study this too, and just to mention, well, those people just come up with the dumbest names because they're all weirdos. So here's some names like Reaper, Grim, Sickle. Great job, you guys. Those are the genes names in fruit flies that are involved in programmed cell death. Because the Drosophila geneticists are just like, like that. You're just like, go home. No, go back to your lab. Go back to your lab. We don't need to learn any of these names. Look at these arrows. Just BCL2 and caspase. Understand that you could learn more, in some advanced course, but it would just take a while to sort it all out. What's going on here? Tremendous amounts of careful regulation. We want the cells to be poised to go through apoptosis, but only in the right moment, And there can be a lot of different moments where we want them to go through programmed cell death. So we have the term ortholog. It's different. Orthologs are between species. Paralogs within a species, orthologs between species. All right, so here's BCL2. I'll just read you this from Wikipedia, just a little bit. BCL2 is the founding member of the BCL2 family. They regulate programmed cell death either by inducing pro-apoptotic or inhibiting anti-apoptotic apoptosis. BCL2 is specifically considered an important anti-apoptotic protein, but is not considered a proto-oncogene because it's not a growth signal transducer. So now it's saying right away, it's starting to say this thing is involved in cancer, but in a different way than you're used to thinking, because you learned about oncogenes like RAS, and this is different. It doesn't stimulate growth. It doesn't regulate growth at all. It's involved in apoptosis. Yet, apoptosis was discovered to be something that was important in cancer in addition to out-of-control growth. out of control growth plus a failure to go through apoptosis. So that's Wikipedia explaining that. Yep,
What happens if if BCL2 is inhibited?
then the cell dies.
Okay.
Yep. Okay, so let me just read you this. You don't have to see this, but it's just a really nice paragraph. The story of BCL2 began with the discovery of the gene's involvement in chromosomal translocations found in non-Hodgkin's lymphomas, okay? And I've heard of that kind of cancer. Non-Hodgkin lymphoma is a rare cancer, but I've heard of it. So they're talking about people who have that cancer, and this scientific group led by this Croce discovered that there was a chromosomal translocation. And I talked about that on Tuesday. The chromosomal translocation is going to involve the BCL2 transcription unit that will code the inhibitor and a regulatory region. And what we're going to do is we're going to join in blood cells the regulatory region for an antibody gene to the transcription unit of the BCL2 gene. And we're going to make way too much normal BCL2. And the cell won't die. Right? The cell is not good. That cell's not going to die because it has way too much of this inhibitor. You can't overcome it. It just says, don't die, don't die, don't die. Inhibit, inhibit, inhibit. There's way too much of it. It just makes too much of it. So that's what happens in non-Hodgkin's lymphoma. Not
What
the
was
only
the regulatory region?
for an antibody gene.
Okay.
So in our blood, we have red blood cells and white blood cells. The white blood cells are the immune system. The white blood cells are composed of B cells and T cells. The B cells make antibodies, which we've heard about in COVID, like that, right? Those antibodies are proteins. So in the B cells, there must be antibody genes. And a B cell makes lots of antibodies, so the regulatory region for an antibody gene is very strong. And in B cells, there are transcription factors that bind to it and make a lot of activation. Lots of RNA, lots of protein. And we bring that regulatory region to the BCL2 transcription unit, and in those cells we make way too much. At first, in a person, that's not a problem, right? The cells shouldn't die, they're fine. But it's going to contribute to a problem because the cells are going to get other mutations where they start to grow out of control, and they should die, but they won't because they have a ton of wild type normal BCL2 really strongly inhibiting the pathway. So let's just read on. It says most low-grade non-Hodgkin's lymphomas contain this translocation. And the low-grade means the person really doesn't have cancer yet in their life. There might be some detection of some problems, but maybe not. And maybe for 20 or 30 years they have this issue. Like that, just a little issue. They don't really have cancer. It's because it's not really growing out of control. Like that. So they already have this specific translocation, and they describe it, driving high levels of BCL2 expression in these B cells. These non-Hodgkin's lymphomas are termed low grade because the B cells accumulate without having a high proliferative rate, which is an early clue that BCL2 operated in a manner distinct from these oncogenes like MYC and RAS. It prolongs the lifespan rather than stimulating cell division, like that. And then blah, 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 and it probably says down here that they get, more mutations, like that, okay? So that's the cancer that we're focused on here, right? We talked about a chromosomal translocation of the woman with skin cancer. It was a different gene, right? Okay, this just shows it. You could see that under a microscope. They've known it for years. They call people who have non-Hodgkin's lymphoma in part because they look at your chromosomal translocation. We found it under a microscope. Okay, so then I just want to read down here. This is further down on the Wikipedia page. It says cancer. Cancer can be seen as a disturbance in the homeostatic balance between cell growth and cell death, overexpression of anti-apoptotic genes, and underexpression of pro-apoptotic genes can result in the lack of cell death that is characteristic of cancer. An example can be seen in lymphomas. The overexpression of the anti-apoptotic BCL2 protein in lymphocytes alone does not cause cancer, but simultaneous overexpression of BCL2 And MYC may produce aggressive big B-cell malignancies, including lymphomas. Right? So that's very logical, and that's why we're covering it in chapter 19 in this section about cancer. That cancer is always multiple mutations and often something that causes the cell to grow too fast, and then something that causes the cell to fail to die. Okay, so in these B-cell lymphomas, we got MYC causing trouble, stimulating growth, and then we have too much BCL2, not dying. Yep. Okay. Okay, so
is you said that it can kind of last for 20 or 30 years and not be cancerous, so does it depend on the activation of the MYC mutation for it to become cancerous?
yes. Yeah, and a little bit, I kind of want to not dive into that too much, but our bodies are always producing new blood cells, so our bone marrow is always making new blood cells. Our blood cells are dying just naturally. They just die, and it's like our skin, where we're always growing new skin and our old died skin is rubbing off like that. And so that's how they get this low grade, is that it's not that they're growing too fast, but they can detect it because, wow, these guys have too many of those cells, and it's because they're not doing a good job dying like that. And then you start with that, not a problem, walking around going, yeah, I can run and jump really well. But then you get a mutation in one of these oncogenes, and when they start to grow too fast, you've lost the ability to wipe those bad cells out. Yep. Okay, so another thing we wanted to go loop back on was this P53, just to show you that we talked about that, and we emphasized that in the chapter 18 story. P53 is a transcription factor that binds to the enhancer of P21 gene, and we get inhibition of START. So there's P21, a small inhibitory protein binding to the CDK cyclin complex and just being the school patrol. And just to show you this visual, so here it is later, the DNA damage has not been repaired, and the P53 gets phosphorylated, and now also binds to the enhancer of pro-apoptotic members of the BCL2 gene family. And they cause the cell to die. And it's very much like if you left your lid up on your washing machine, it waited, but after a certain length of time, it just blows up. Right? Normally it would just wait for years. But here's a built-in thing saying, if you haven't fixed this problem, it's not, we're going to apoptosis this cell. Yep.
So I remember last time you were talking about how P53 ends up becoming more and more phosphorylated. In this picture, it just looks like you're getting increased concentrations of P53. Is it like a combination of both of those?
Probably, yeah. I don't know. That's a good question, though. That's a great question. Yep, I don't know.
And just to really say it again, so Puma is gonna inhibit the inhibitor, and then caspase is gonna be able to break down integrate apoptosis.
Yep.
Cool.
All right, so that's that. We talked about that on Tuesday, but the projector was broken. So I wanted to show you some of those images. When I sent out the audio recording, because we didn't have the video on Tuesday, I sent out the PowerPoint slides. So I sent these images out today, because I would have shown them on Tuesday. So we're really just reviewing on Tuesday from the broken projector. Yep, Hannah?
So P53 binds to the enhancer P21.
Yep.
Okay. That's all.
Yep. Okay. So I want, at the end of class on Tuesday, we talked a little bit about stem cells. I just want to show you this stem cell biology. And we can think of 3 types of stem cells we want to learn about: adult stem cells, embryonic stem cells, and these induced pluripotent stem cells. So that's our list from the second part of chapter 19. Okay, and I just want to show you the first one. Our favorite example of that is the muscle satellite stem cells. So maybe for this exam, you want to go back and watch the videos where we started our course. But maybe you understand that really well and their role. So we'll talk about them a little more. The book talks about muscle satellite stem cells in chapter 19. We should be experts like that. We want to talk more, a little bit more about them and these other types of stem cells. Before we do that, I thought, because our exam is 1 week, I thought I'd just tuck in here, insert in here today, some about chapter 20. Because chapter 20 is a really important chapter. I hope a lot of it you'll see as being like, wow, this is a good chapter for you to read. Because it really stacks up everything we've been trying to stack up in our course. So I'm just inserting this, and then we'll loop back to the stem cell piece. Maybe we won't get back to that till Tuesday, but I want to tuck this in and talk, for half an hour about this. Okay, so here's one way to organize a big part of chapter 20, not all of it, but part of it, and it's satisfying to see this. Here's the 4, I think, and the book doesn't really say this directly, but I think it's a good way to think about this. There are these ways that scientists really started learning about molecular bases of cancer, right? Cancer is caused by mutations, but what genes? What's it doing? Godwin, do you have a question?
I'm in chapter 20 now, right?
Yep. Yeah, and trying to give you some, some navigation for that. Okay, so the first one, this is where we're going to be tested on this a lot. Our book explains this in more detail, and we should just understand this just right away. RAS. Talked about that a lot. Our book author career. Okay, so oncogenic retroviruses back in the 1970s, there were just a couple dozen of them, none in humans. They were just normal retroviruses out in the nature that could cause infections in some cases in a cat, or a different one in a mouse, or a different one in a monkey, or a chicken, like that. And scientists had found that certain variants or mutations or versions of those retroviruses could cause cancer upon infection. And they discovered it was because they had picked up one of the host genes. It wasn't the virus that was causing a problem, it was a host gene that they'd accidentally picked up. So we talked about that a lot, and that's a big part of this, okay? So the book in chapter 9, I guess that's really from the previous edition, but chapter 20, and we've shown this before. There's the regular wild type normal retrovirus, and there's the oncogenic one, and it's picked up a host gene, in this case a gene called SARC, and that SARC is the problem. Okay, so that's that, but you really need to understand that like the back of your hand. Then this is a second one. It's a different thing. It's not the oncogenic retroviruses, it's some other viruses. And there's some other viruses that naturally cause uncontrolled proliferation, or they repress programmed cell death, and they don't cause cancer directly, but they set the stage for cancer. And if you think about it, it's not surprising for a virus to cause stimulate growth. Because when viruses infect, they want to replicate. And lots of them are DNA viruses. So they would really like it if the cell was in S phase. And it had a lot of DNA polymerase and a lot of AGCT like that the virus could use. Right? The virus is trying to take over the cell to make more of itself, replicate. And then the cell might try to defend itself, and the cells do. When they get infected, they try to go through apoptosis. So the virus could try to block that. And chapter 20 talks about that. So here shows a list, and we don't have to memorize this list. Like that. But here's one we'll talk about, a papillomavirus that causes cervical carcinoma. And you don't need to write down that big long word because it's HPV, which is pretty well known in the general public. HPV is a sexually transmitted disease that causes something called genital warts. And there's a vaccine that blocks it. And folks are encouraged to get that vaccine when they're children because it will block the HPV infection and genital warts. A wart is like a benign growth. It's not cancer, but it's a growth. And that's an indication that the cell is a little bit out of control, but not causing an aggressive cancer growth like that. And so this just shows that here's the HPV genome, and it makes some proteins, and this protein degrades P53. P53, when there's DNA damage, can help prevent the cell from going into S phase. And this virus would like the cell to go into S phase, not for the cell, but for the virus. Because the virus is made of DNA and it wants to replicate. And it needs help from the cell replicating. So it would like to just get that cell moved into S phase. It doesn't care if that's going to endanger the life of that cell or organism. It just would like to move it in there. Okay? So isn't that, it's just crazy to see that, right? Because you probably have heard of HPV, and when you get down to the molecular level, you just shrug and say, well, that's the problem. And then there's a second problem. Here's another viral protein, and it inhibits RB. And, you know, we should, maybe you don't understand P53 or RB well enough yet, but when you see that, you should just go like, oh, crap. Because RB is a transcription factor that's a co-repressor E2F. And E2F is bound right now on the enhancer of genes like DNA polymerase, right? You know, we just like to get you to make some DNA polymerase and other genes that are for enzymes that stimulate the synthesis of the AGCT, like that, you know? So it's just like molecular biology is like that, where it just gives you the shivers because of its cause and effect relationships, like that. So from this list, it would be great for us to recognize that. Not all these, but HPV. And then see HPV, what does it do? I'd be able to say on a test, you know, what does the HPV do that's a problem for the cell? And the answer is it causes P53 to get degraded and it represses RB, right? So it's like that where we're kind of pulling our learning together, right? Because P53 is from chapter 18 and 19, and RB is from chapter 18, like that, you know, and we're like, oh, dang, right? So chapter 20 is a lot of this, and that's why I wanted to jump into it, because at first you might be like, oh, there's a lot in chapter 20, and there is, but a lot of it is just pulling it together in ways that, you know, we need to understand this material well enough to be like, oh, holy cow. Yeah?
Are the viral proteins E whatever? Is that what that is? Okay.
Mm-hmm. Yeah, so here's the DNA for the genome for the virus.
Oh, for the
And
virus.
yeah, that's the virus's genome. And so these would be, there would be genes in the virus that it brings when it infects the cells, and then they get expressed, and these would be the proteins. And it's showing, you know, oh, these proteins, you know, it makes some different proteins, but these two it wants to show, right? How do those help the virus? Why does the virus have the E6 and the E7, E1A gene? The answer is it evolved that to try to get the cell to move into the S phase, because that's going to help the virus. The virus is like, I'm looking for some DNA polymerase, looking for some help here. Crank it up a lot, right? Viruses just want to crank it up, and there's a huge evolutionary pressure, natural selection pressure. If they could get the host cell to help them replicate more and faster, that would be good. So there's nothing mean about this. It's just virus is trying to get its job done, which is making lots of copies. Like that, yeah?
Can you repeat, excuse me, the significance of the P53 degradation?
Yep. So P53 is this transcription factor that gets activated when there's DNA damage. And it binds to the enhancer of the P21 gene and stimulates its expression. And P21 blocks the cell at start. This will just help encourage the cell to go through start and get into that S phase, where the cell would be making the machinery for DNA replication. That's what the virus wants. So let's move it forward. Get into that. It's not trying to help the cell in any way. It's trying to help itself. And it would be helpful for the virus if it got the cell into S phase. It doesn't care if the cell's in S phase as far as copying it, the chromosomes in the cell. Just would like to get the machinery for DNA synthesis going. Let's get that going.
But I thought by repressing RB, you're not activating the E2F genes.
Yeah, so this is an interesting thing, right? Because it gets, right, remember start is complicated. So you're right, I'd say that the light stays green, but remember that what RB is repressing is the E2F, which is activate the synthesis of DNA polymerase and AGCT. So in that sense, it's not even getting the cell into start. It's kind of getting it off to the side where it's getting the machinery going for
But
replication.
then the E2F genes aren't being activated, so you're not producing the AGCTs, and you're not.
You are. So this is where you have to understand that well. So the gene for, let's say, DNA polymerase,
Yep.
it already has E2F bound to it,
Yep.
and it's trying to activate it. And the role of RB is to repress that. So if we block RB, we'll let E2F do its job. And it's true that E2F does not really turn the light green, but I'm saying that because it starts activating the stuff that's S phase stuff that the
Gotcha.
virus wants.
I
So
thought,
it's complicated. So you see, that's this thing where we have to tie that learning together. So this is kind of like a good, kind of like a reflection of like, do we understand that E2F function? Yep.
yeah, I guess I thought that if RB was activated, it was not bound to the E2F, and if it was repressed, it was staying stuck to the E2F. So that makes more sense.
Yes, RB is, when it's active, it's a co-repressor of E2F,
Yeah.
and we're going to block that. That's the thing, again, gets back to that kind of the inhibition of inhibitor, double
Yeah.
negative thing that's a little bit complicated to soak up. Yep,
Yeah.
yep, Catherine?
Oh, that's what I was just gonna say.
Yeah, yeah, so it takes some thinking and talking out loud and maybe riding your bike around a little bit and being like, oh man. But expect that you're gonna understand it, right? Because it is like, parts of a tractor thing where you're like, okay, I see how these fit together, right, like that. And so here's EPV, and I'm just gonna try to read some of this. That's the virus we wanna focus on. Is this an oncogenic retrovirus? No. Oncogenic retroviruses, first of all, are retroviruses that have RNA genomes, and oncogenic retroviruses have a mutant or defective host gene. This is a different story, right? This is a different story. It's a DNA virus, and it doesn't cause cancer, but it does cause trouble. It causes these genital warts, which is not surprising that there would be some kind of growth of tissue as a result of this infection. Not a wild cancerous growth, but just sort of too much growth there. What's going on? And the answer is this virus trying to replicate, and it's not trying to stimulate the cell to grow and divide, but it pushes it in to try to get into S phase. Brooklyn, do you have a question?
You said it's a defective host gene. What about it is defective?
So
What's that?
this is not an oncogenic retrovirus,
Oh.
right? So I'm just going to go back to this. So see, we've talked about that, and this is not that.
Okay, so the oncogenic retrovirus is the defective host gene?
Yeah, the oncogenic retroviruses accidentally picked up a mutant host gene.
Gotcha.
This isn't that. This is just a virus, right? It doesn't cause cancer. It causes genital warts, which is not cancer. But in doing that, you see what it's doing. It's trying to replicate itself, and in doing that, it pushes into the cell into dangerous territory because it's pushing it, trying to get it past that start. Maybe the light turned green, maybe it doesn't quite turn green, but it's pushing to try to get it to that place where the cell will have DNA polymerase and AGCT. Okay? All right, and so we're just learning about that one. There's a list of them. We're learning about that one. That's a good one. And so here's just some writing about HPV Wikipedia. Human papillomavirus infection, HPV, is caused by HPV. Most cause no symptoms and resolve spontaneously. Some people, an HPV infection persists and results in warts or precancerous lesions. These precancerous lesions increase the risk of cancer of cervix, vulva, so forth cancer. Like that. Nearly all cervical cancer is due to HPV. Like that, right? So there's that. And then there's this encouragement, and some say HPV vaccines can prevent the most common types of infection. The most effective, they should be used before the onset of sexual activity. And so there's a controversy in our country about giving a vaccine to prevent sexually transmitted diseases to children. Because people in the general public say, well, isn't that sending a bad message to kids about sexuality and whether they should be sexually active? And this is Wikipedia, you know, just going tink, 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 tink. And we see why you want to prevent the infection. Because the infection leads to the degradation of P53 and the inhibition of RB. You know, it just leads to that danger like that, okay? HPV is the most commonly sexually transmitted infection globally, like that. You know, it's a huge topic like that, right? Yet, you know, you've heard about it because it's somewhat controversial. Should it be? No. Yep.
What is different about the HPVs that cause no symptoms and resolve spontaneously? Do they not inhibit RB, or do they not?
No, they do. But inhibiting RB doesn't cause a cell to be cancerous. It doesn't stimulate just uncontrolled growth. What it does is it encourages the cell to get into close to S phase or get into S phase.
So there's other stopping mechanisms that are like, maybe don't do that?
Yeah, right? We haven't damaged anything else, you know, and there are many controls. That's a good question because the start has all kinds of checks and balances. So we're, you know, pushing ahead on a couple of them, you know, a couple of weak points, but not enough to just make the cell grow, grow, grow. And the idea that they're asymptomatic, no symptoms, it's just like, you know, some viruses cause like, cold, you know, and there's some symptoms there, but it's not like it's going to cause you to die, right? So HPV is just a minor infection like that. But because the nature of the infection is to try to let the virus replicate, it just happens to set the cell up into dangerous territory, where it says, you know, some people then continue on in their lives, and at some point they get some genital warts. They get a little bit of, you know, growth, precancerous growth, like that. And it's like 10, 20 different lengths of time, right? Because they've had the infection, and it makes them vulnerable, right? If you combine somebody who has an HPV infection with somebody that gets another mutation, gets a mutation in one of their genes, well, they could be in trouble, right? Because there are lots of checks and balances, but having had that infection removed a couple of the checks like that, you know?
So are you saying like they could have the infection at one point and then like 10, 20 years down the line they could be more vulnerable to like cancer?
Yeah.
Okay.
Yeah, and it's these people who had the infection and then it, I'd say, doesn't get resolved, doesn't get, you know, it is not producing any symptoms, but some people just have the infection for a longer time, just don't get rid of it. So it's there kind of like lingering, and they get growth of some genital warts that's just like a growth that's not cancerous, but it's like, why is that growth happening? And the answer is because their cell cycle has been messed with by this virus like that, you know? And it just sets them up so that if you have a mutation that causes loss of another, checks or balances, you know, like that, just more likely to, you know, lead to trouble. Yeah, years later. Yep. Yeah. What's it say? It says something. Where's that 70% thing? It says something. Oh. Yeah, nearly all cervical cancer is due to HPV. Right? I've heard of people that had cervical cancer. Right? Like like that. I think my grandma had cervical cancer. I think. I mean, that was a long time ago, so, you know, their diagnosis was probably lame. But yeah, you know, and you're like, you see statements like that when you get down to this molecular level, you're just like, it's just like, wow, right? Like that, okay? So that. Then here's just a little bit about genital warts. Wikipedia: genital warts are contagious, fleshy out growths in the genital or anal area. They are one of the most common types of sexually transmitted infection, also known as venereal warts or something. They're caused by HPV. So again, it's not causing cancer. It's not even causing genital warts in most people, because it's the most commonly sexually transmitted disease, but doesn't have any symptom. But in some people, the infection kind of lingers, and they get some growth that produces these warts, these little growths, and that can lead to cancer if they get mutations that lose some of the checks. Yep. So, you know, just lots of Wikipedia pages about this, because it's a huge topic, and then it's controversial, you know, so you hear about it, and it's curious and stuff, but at the medical and certainly at the scientific and molecular level, you're just like, oh my goodness. So you'll show you that. And our book talks about this in chapter 20, right? So it's just one of the chunky things. The book is just rolling through going, so you guys remember P53 and RB, here they are, getting messed up, you know? So you just have to study that this weekend and think about that. And you know, just some Wikipedia sites about controversy of the vaccine, right? Just interesting like that. So our course is not about the controversy, but you know, just to mention it, because that's a part of the news in you know, the United States. Okay. So the third one, that's when we did all the learning. Our book, the book talks about it in this chapter, but we've done our learning, and we have a really good example on an exam. Especially the final exam, I could say, well, what are the 3 good examples of chromosomal translocations? And you'd be like, well, that woman that had skin cancer had a translocation between the skin collagen gene and the PDGF gene. Then we saw Burkitt's lymphoma, and that's a translocation between the MYC gene, transcription unit, and one of the antibody genes. And then we saw BCL2, and that's a translocation between BCL2 and one of the antibody genes, like that. In all 3 cases, they're really good examples because they're true and examples of specific cancers that are caused by them. Then they're in certain cell types because that's where the regulatory region is active. The skin collagen is active in skin cells, so could contribute to skin cancer, right? Couldn't contribute to cervical cancer because skin collagen gene is not made in cervix, like that, you know? And then BCL2 and MYC, those are lymphomas, cancers of the blood, because it's those B cells that normally make antibody that are making too much normal MYC or too much BCL2. And so here in chapter 20, the book rolls through this, but it should be really familiar territory to us, but it should be solidifying that. It's like review and stack, you know, review and stack, and there's a lot of that in chapter 20. Review, get solid on that, stack a little more knowledge on there, like that. And this is just that visual that we've talked about before. MYC, there are actually 4 different antibody genes, so MYC could be translocated to different ones like that, okay? Then the last one is this gene amplification. We're also good on that. Our one example is MYC, where in some cancers, MYC has been amplified. And it just means that we accidentally made copies, many copies of it, just in that cancer cell. And so here's just a visual. Normal growth control, our example would be MYC. Then gene amplification, just too much MYC. MYC protein is really carefully regulated. We try to make it unstable. We make the RNA unstable. We make the protein unstable. We make it hard for the ribosome to synthesize MYC. And if we have gene amplification, you get too much MYC. And that's found in some cancers. The MYC protein is normal. No mutations in the MYC protein. It's just that we've made several copies of the gene, and we're making too much MYC, and so it overwhelms the regulation. How many? Like in some cancers, there'd be 100 or 150 copies. You know, just because, you know, cancer is just growing out of control and it's getting problems with, you know, repairing DNA and stuff like that. Then this thing, it just says, you know, that gene amplification, there's no mutations within the gene. It's just that you copy the gene, so you're going to get too much of the more normal gene product. And it just compares and contrasts this where you could have mutation in the gene, and you could have a mutation within the transcription unit, make a defective protein like what? Like RAS, or in the regulatory, make a mutation that is mutation in the regulation of the gene, like that. Making too much of it for an oncogene or too little bit for a tumor suppressor. So that's that gene amplification. And so that's a nice list. And I say these are ones that scientists still study a lot. But over the last 50 years, these are examples that have really helped scientists understand molecular mechanisms of cancer, right? The oncogenic retroviruses back in the 70s, then these other viruses that can lead to cancer, like HPV, they chromosomal translocations of either oncogenes or apoptotic genes, and then gene amplification of oncogenes. Yeah?
You just said something about making a point mutation. Is that unrelated to gene amplification?
Mm-hmm. Yeah, that's a good question, and this is a good figure because it's emphasizing this is one thing, and it's amplifying the gene. Too many copies of the gene, no mutations in the gene. This is a different gene, different story. So RAS involves point mutations in the coding region. It wouldn't really matter if you amplified RAS, because it would just be normal RAS, and too much RAS is not a problem. But a defective form of RAS is a problem. Yep. Okay, so that's that. And we're going to stop and have a concept chat. So we haven't had one of those for a while. Here's the book, just laying its outline out. I just want to show you that there's more to say about chapter 20, more to learn. But that's one way to organize some of the learning, a big chunk of the learning, but not all the learning we need to do is like that. And I think it's a good list, those four for us, because we know quite a bit about most of them, and it was really just HPV that's new to us, but the story of it is like, oh my gosh, familiar, okay? So, you know, you can see that we just talked of some of these, but let's pause, have a concept chat, and talk about three things that you think were important that we talked about today. Oh, I got to set up a attendance sheet.
Oh,
I really enjoyed that. And same with like caskets. And then the parent lots would be within the organism. Not what
I said apoptosis is how I I was going to say simpler. I'll probably draw it, but it's like, I was like, this points at this, and then this points at this.
Did we say that nematodes have like a BCL2 like gene?
Ortholog?
Ortholog?
Mm-hmm. Definitely, yep. Just one family member. Just one.
Not the
An
whole
only
thing.
child. Yeah. They're really simple. They only live for 2 weeks, you know? It's just a, much simpler relative of ours. Yeah. Yep. We're going to do this for another minute, and then we'll check in, and we'll have a couple minutes to talk a little bit more.
And I guess it's nice what you wrote is you can yeah, so I like the course.
It was then the woman with skin cancer. The woman that has skin cancer, this is skin collagen, and this is the cancer that was tied into BCL2. Okay, Sophia, what is one thing that your group talked about today? Touched on everything? What's one thing?
I talked about
HPV?
and about the BCL2 and how that relates to apoptosis.
Yep. BCL2 is inhibitor of apoptosis. And then there's the BCL2 gene family that codes for several proteins, some of which are inhibitors, like BCL2, anti-apoptotic, some of which are activators of apoptosis, pro-apoptotic. Awesome. Catherine, what is one thing that you thought was important today?
We just talked about like the 4 different mechanisms that we talked about with cancer, and just kind of like a general overview of the oncogenic retroviruses, those other viruses.
Yeah, so this is a list that I just made up to help organize our learning. Our author doesn't have this list. A lot of times in class like that, when the instructor makes an organizing list, it's an example of how a person could study and learn through the stuff, is to try to organize it based on some of the stuff you are familiar with and some new stuff. Brooklyn, what's another thing that your group talked about that you thought was important?
We also talked about that list that you gave us, the 4 most common ways scientists learn about cancer, molecularly,
Yep. Yeah, that's a new one for us, and that's something that there's a really good chance you've heard about just in your personal life and your healthcare. You probably good chance you've heard about it, and you can see that for a molecular biologist, it's like there's nothing else that's interesting about HPV except that the HPV genome codes for proteins that lead to the degradation of p53. Because we know, as molecular biologists, what the role of is for p53. And so it's like, oh my gosh, that's really striking. And then another HPV gene codes for a protein that inhibits, inhibits RB. And we're like, oh my gosh, right? You don't need to know the names, of those HPV proteins, but we really need to understand p53 and RB learning, and then tie it to HPV. With that, yep, yeah, yeah, Joe.
I just have a quick question. You were talking about, it was earlier in the lecture, I think it was when you were talking about, it might have been when you were talking about non-Hodgkin's lymphoma, or like one of the types of cancers. You were saying that they want the cell, it's when they can control the cell cycle, and they want the cell to be in S phase. Is it because like they have the most material available to them when they can control the cell cycle and have it in S phase, that they have like, I guess like the most supplies that they need to like feed uncontrolled cell replication?
Right. Yeah. Yeah. When a cell is in G1, so an uninfected cell is in G1, and it's in G1 most of the time, it has just a little bit of DNA polymerase that's there in case there's any DNA damage that happens during, like from UV light, and we're going to just need to do a little repair. And it's just a little bit of DNA polymerase. And for a cell to be in S phase and replicating all of its very long chromosomes, it needs to have a lot of DNA polymerase, a real lot. And so when we say the light turns green, that involves really just stockpiling and activating that replication process. So the cell will say, we are going to phosphorylate the MCMs to show DNA polymerase where to start replicating the chromosome. We're also going to make way more MCM, because now we really need it. We had a little bit around, but now we need a lot. We're going to make a ton of DNA polymerase. Yeah. So that's what's going on there, and it's because this is a DNA virus.
Yeah. Okay.
Do retroviruses do that? No, because they're RNA viruses. They want the cell in G1 because they want RNA polymerase to replicate their genome like that, right? And so it would be a DNA polymerase that would be like pushing it into that S-phase status where they're really stockpiling and synthesizing the nucleotide. You mentioned non-Hodgkin's lymphoma and BCL2, so that's a completely different story, right? Right, that's an earlier story about the role of BCL2 and how in non-Hodgkin's lymphoma we actually make too much of it. Just to clarify that a little bit more, there's some confusion there because in both that BCL2 non-Hodgkin's lymphoma story and the HPV story, we did talk about how there's a kind of an extended latent phase where the person does not have cancer, but they kind of are in a dangerous situation. And so with non-Hodgkin's lymphoma, there's this period where the doctors can tell they have too many B cells, and it's because they have too much BCL2 and the B cells are not going through their normal cell death. And so there can be this long latent period where they are like, we're concerned and we need to keep track of this. And then suddenly they get a mutation like in MYC, and they start really making too many of the B cells, and then they're not dying and they accumulate really fast. Then similar but very independent and separate from that is the situation with HPV, where a person get HPV, they get infection, it goes away, they're fine. Some people have a longer lingering HPV infection that leads to some growth because they're kind of pushing into that stockpiling scenario, and they start to get some very slow growth and the appearance of some slow growing warts like that, genital warts. And those genital warts are appearing for that specific reason. A person can have warts elsewhere on their body like that. Warts are not necessarily a dangerous thing, but they are slow growth. Moles also are an unusual kind of growth, and we want to watch that because it's what if it grows and starts growing more. So a person that has a HPV infection can have a very long time, many, many years, where they might have genital warts and really no problems. But if they pick up a mutation, they've already lost some checks and balances, and now they're just rolling forward with cervical cancer. So I think that's where you could get confused, and we just really need to, really separate those because they're really different stories. Other questions people have?
I have a question. This isn't really related to what we kind of talked about in this class, like not in this class, but like today. In the textbook for chapter 19, it kind of shows the cell cycle, and then it discusses the fluctuation of different cyclins and kinases. Do we need to, or CDKs? Do we need to really like know when the combinations are happening in the cell cycle, like specifically when like the combination of like cyclin B and
No.
CDKs? No.
So the cyclins are a gene family, paralogs that arose by gene duplication, and the genes are named by letters. So there are different letters of cyclins, and we don't need to know the different letters. There are different cyclins at G2M versus G1S, but we don't need to know that. Then the CDKs are also a gene family, and the different genes are named by numbering, and we don't need to know the numbers. There are some facts we need to know, like for what's happening at the G2 to M transition. There are already plenty of cyclins. Over at the G1 to S, we need to know that lots of times there aren't enough cyclins, and we need to stimulate the cyclin gene to be expressed and make more cyclins, and that's the rate limiting step. But we don't need to know the details of which cyclin and which CDKs. Let me just show this and show you that in chapter 20, there's talk about cancer, and if you took this course and said, oh, we're going to learn about cancer, this is what you thought you were going to learn about. And we want to learn a little bit about this, but it's really the medical aspect of cancer. So there's some highlights like, oh, there are only 3 main types of cancer, like that. And so don't get derailed by that, because if you want to go to medical school or whatever and you find that really appealing, don't get derailed by that. There's some of that we want to learn, but the big part is this molecular parts of chapter 20. And then the last third of the chapter is about therapeutics, and that's where CAR T-cell therapy comes up, that's where tamoxifen stories come up, that's where cetuximab stories come up. And so that's what we really want to drill down. So on Tuesday, I'll talk some more about chapter 20 and answer questions about it, like that, as you try to pull stuff together. And then there's more we want to talk about the stem cells, but we should be able to fit it in and have our test a week from today.
Will all of chapter 20 be on this?
Yep, yeah, all of chapter 18, 19, and 20. Yep, yeah, those are really good chapters with lots of good learning we can do there. So, yep, that's where we're at. And then I just handed out the sign-up sheet, so find that if you didn't find it, sign up. And great. Hi.
Can we meet sometime, like maybe Tuesday after class to go over some material, or maybe not
Sure.
Tuesday, but Wednesday actually? Do you have time on Wednesday?
Sure. Yeah, and Hannah's talking about meeting. You can send me emails about questions, like that. I'll be looking for questions, or we can set up times to meet. Yes.
Yeah, because I was thinking like Wednesday we could like meet. You might know how like last time we met in your lab and we just like went over some stuff that was super
What
helpful.
time?
I get out of class around 1:45, so anytime after 1:45 I can meet.
Okay, so let's meet then, like right after class. Let's say
Like 1.
2.
Okay.
Okay, 2 on Wednesday.
Yep, sounds good.
Okay,
All right,
cool.
see you then.
Yep, great.
Could we? I might email you for a meeting.
Sure, that sounds great. Yep.